Hi, my name is Arafas, and today we're going to talk about the article, The Heterologous Expression of the Iris lactea type 2 Metallothionein ILMT2B gene and how it enhances copper tolerance in Arabidopsis thaliana. So, Iris lactea is a plant that is a hyperaccumulator of copper. This is done by the metallothioneins present, and they have a high content of cysteine, so cysteines bind to metal cations. First, the sequence was isolated from the iris lactea by RT-PCR, and it was compared to other sequences of MTs to determine the type. So the motifs present in the amino terminal end were congruent with the type 2 metallothioneins. So it was then compared to other MT type 2 to determine the similarity between them. It was found that they were similar in a range between 49 and 65%. The closest phylogenetic relationship was with MPMT2, which comes from Narcissus plants. The gene transcription rate was determined by exposing the plants to a high concentration of copper and measuring the transcription at different times. So at the 6-hour time mark, it was found that the highest concentration of the transcript was present in the plant in different sections of it. So we can say that the transcription of this gene is induced by extracellular concentration of copper. After this, the coding sequence was introduced into a plasmid pCambia, and this was in turn introduced in Arabidopsis thaliana, and the transgenic plants were selected by an assay, and we can see that the wild type doesn't have this gene. This was a gene which served only as a control. So after this, the root length was compared with different concentrations of copper. The control group didn't have any copper, and we can see that the root lengths are the same for the three types of plants. After this, 550 micromolar copper concentration was added, and we can see that there is a significantly higher root length in the transformants than in the wild type. This effect is also seen at a higher concentration of copper. So we can say that the presence of this MT enhances the proper development of root systems. After this, hydrogen peroxide production was measured. Hydrogen peroxide is produced when a plant is stressed out by copper. So in a time where there is no copper in the medium, the same amount of hydrogen peroxide is produced by the three plants. But if we increase the copper concentration, we can see that the hydrogen peroxide increases significantly in the wild type compared to the transformed plants. So we can say that MTs act as antioxidants against reactive oxygen species. So that's all for this, and thank you for watching.